So, given the cellular mechanisms of long-term potentiation and the anatomy and the wiring diagram of the hippocampus, some people, particularly a scientist called Edmund Rolls, have come up with a plausible and very influential model of how the hippocampus works. So let me take you through roughly how that model works. I'm going to start by reminding you of an autoencoder. And you remember we said that what the autoencoder does is enables us to recover from a partial memory all the rest of the context. So using these recurrent connections, so each cell excites all the other cells through a hebbian synapse. And so if we're trying to learn this sequence, for example, and we train on this, we learn, but then we present it with a missing cue, what we get is a recovery of the original idea, so the original string. So for example, we said we want to associate mouse with cheese and with trap. So if I say to you cheese and mouse, you will automatically think of trap. So here I'm just saying cheese and mouse, and here coming up with trap as well. Now let's also relate that. Let's try and relate that now to what we've been saying about the hippocampus. So here's a rough structure again of the hippocampus. We've got the, to make a slightly bigger dentate gyrus. We've got the dentate gyrus here, and then we've got CA1. Let's make that bigger too. We've got CA3, and then CA1 and the subiculum. And we said, didn't we, that we've got the perforant pathway comes in from the so the perforant pathway comes in, synapses onto mossy fibres of the dentate gyrus. These then synapse onto pyramidal cells in CA3, and these send a collateral through the Schaefer collateral these synapse onto CA1 cells, and these go to the subiculum and out. And we also said that there's this direct pathway from the perforant path onto these cells as well. So here's the story. If you look at the synapses here, what you find is in this synapse here, we find that these are few in number but strong. If you look at these synapses here, these are many and weak. So that's observation number one. Observation number two is that these pyramidal cells make lots and lots of connections with each other. just like here. So here's the idea. Basically, the model is that this region, so CA3, is an autoencoder because these pyramidal cells are weakly connected to each other, and the job of CA3 is to form a temporary memory buffer associating different inputs. That can then, through back connections here back to the entorhinal cortex and then back to the rest of the brain, the cortex if you like, that can then recover associations in the entorhinal cortex that originally caused this association down here. So basically what CA3 will do will be to repair this association and then send it back to the entorhinal cortex. So that's our autoencoder. But there is a slight problem. Imagine that in a different context, you wanted to associate cheese with toast and sandwiches. How do we deal with that? Because now we've got two sets of associations. We need to separate out these in some way, and that is the job of the dentate gyrus. What the dentate gyrus does is it separates out the input space by taking in a small number of features and then multiplying them, distributing them across lots of different neurons, and then recombining them in different ways. So that's what, according to the Rolls model, that's what the dentate gyrus is trying to do. So let's look at that slightly in a somewhat cleaner way. We've got the entorhinal cortex with input from the temporal lobe. The entorhinal cortex then sends a fibre down and it interacts with, in the dentate gyrus, with a mossy fibre. We also have CA1 neurons and this dentate gyrus, this entorhinal cortex input also affects those. Meanwhile, the mossy fibres also synapse onto the CA1 cells, and so what's happening here is we've got recoding of the input designed to produce a separation of the different contexts. Sorry, I've realised there's actually a mistake here. This is, of course, is CA3. CA3. So these CA3 neurons, what they do is they auto-associate, and that produces the autoencoder. These outputs then go to CA1, a different colour for those, blue, and that provides the output back up to the entorhinal cortex. Similarly, so what happens then, if we've got a partial input coming down from the entorhinal cortex, the first thing that happens is that the autoencoder will try to form the associations and then send them back up to the entorhinal cortex to complete the memory. Over time, the dentate gyrus learns to separate things out so that the autoencoder can work effectively in different contexts. So as I say, this is a theoretical model, and if you look up, there's some very detailed papers by Edmund Rolls and colleagues. You will find them quite difficult to read, but between them and finding tutorials on the web, you can look at this in a little bit more detail. But what they did was to model this using simple on or off neurons, and we're able to simulate quite a lot of the features of the hippocampus.